Hello everyone, I am Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we will go through each topic on the ASCP lecture list. In today's video, we will be discussing intrinsic defects leading to increased erythrocyte destruction. The main function of RBCs is to transport oxygen to the tissues within their 120-day lifespan. To accomplish this task, RBCs require functional enzymes for several purposes, such as maintaining glycolysis and cell membrane shape, preserving reduced hemoglobin iron, protecting hemoglobin and other proteins from oxidative damage, and salvaging nucleotides. Deficiencies in RBC enzymes can impair these functions to different extents and shorten the cell's lifespan. The most crucial metabolic pathways for RBCs are anaerobic glycolysis and the pentose shunt. Glucose 6-phosphate dehydrogenase and pyruvate kinase deficiencies are the most commonly encountered RBC enzymopathies, while other enzymopathies are rare. Glucose 6-phosphate dehydrogenase deficiency. RBCs produce free oxygen radicals and hydrogen peroxide during metabolism and transport, which can cause oxidant stress. Oxidant stress occurs when RBCs are exposed to high levels of oxidants, which can denature hemoglobin, membrane proteins, and lipids, leading to premature hemolysis. Glucose 6-phosphate dehydrogenase, G6PD, is an important enzyme that helps protect hemoglobin and other cellular proteins and lipids from oxidative denaturation. G6PD catalyzes the first step in a series of reactions that detoxify hydrogen peroxide formed from oxygen radicals. G6PD generates reduced nicotinamide adenine dinucleotide phosphate, NADPH, by converting glucose 6-phosphate to 6-phosphogluconate in the hexose monophosphate shunt. NADPH is used to reduce oxidized glutathione, GSSG, to reduced glutathione, GSH, and NADP. Glutathione peroxidase uses GSH to detoxify hydrogen peroxide to water. G6PD deficient RBCs are particularly vulnerable to oxidative damage and subsequent hemolysis during oxidant stress because they cannot generate NADPH effectively. The G6PD gene is found on the X chromosome and encodes the G6PD enzyme that exists as a dimer or tetramer. X-linked inheritance results in males being normal or deficient hemizygotes, while females can be normal homozygotes, deficient homozygotes, compound heterozygotes, or heterozygotes. The activity of the G6PD enzyme in female heterozygotes is intermediate between normal and deficient, as one X chromosome in each cell is randomly inactivated. Therefore, female heterozygotes have a mosaic of RBCs with varying levels of G6PD activity. The proportion of normal to deficient RBCs varies among different heterozygous women due to random X inactivation. Some heterozygous women may experience acute hemolytic episodes when exposed to oxidants, especially if they have a high proportion of G6PD deficient RBCs. G6PD deficiency is a common RBC enzyme defect found in approximately 400 million people globally, with a prevalence of 5%. The prevalence of G6PD deficiency varies by geographic location, with sub-Saharan Africa having the highest prevalence at 7.5%. In the United States, African American males have a prevalence of approximately 10%. G6PD deficiency is more prevalent in geographic areas where malaria is endemic, as it provides protection against the disease. Hemizygous males with G6PD deficiency have been shown to have protection against life-threatening Plasmodium falciparum malaria, while the protective effect is not observed in heterozygous females. A 2010 case control study in Pakistan showed that G6PD deficiency conferred protection against Plasmodium vivax infection, with a greater protective effect in hemizygous males. More than 186 known mutations in the G6PD gene have been reported, with over 85% being single missense mutations. G6PD variants have been divided into classes by the World Health Organization based on clinical symptoms and amount of enzyme activity. Pathophysiology. G6PD deficient RBCs are unable to produce enough NADPH to reduce glutathione, which leads to ineffective detoxification of hydrogen peroxide during oxidative stress. Oxidative damage mainly affects hemoglobin and the RBC membrane, leading to the formation of Heinz bodies, decreased hemoglobin solubility, and precipitation of Heinz bodies. Heinz bodies bind to the inner RBC membrane, causing irreversible damage and loss of deformability. This results in intravascular and extravascular hemolysis, leading to the removal of RBCs from circulation. G6PD activity decreases as RBCs age, with reticulocytes having 5 times more activity than older RBCs. Hence, during oxidant exposure, older RBCs with less G6PD are selectively hemolyzed. Clinical manifestations. Most people with G6PD deficiency do not experience any symptoms in their lifetime. However, some individuals may develop clinical manifestations. The clinical syndromes associated with G6PD deficiency are acute hemolytic anemia, neonatal jaundice, hyperbilirubinemia, and chronic hereditary nonspherocytic hemolytic anemia, HNSK. Acute hemolytic anemia. Acute hemolytic anemia can be caused by oxidative stress from certain triggers including oxidizing drugs or chemicals, infections, and ingestion of fava beans. Hemolysis secondary to drug exposure is the classic manifestation of G6PD deficiency. The discovery of G6PD deficiency in the 1950s was a direct consequence of investigations into the development of hemolysis in certain individuals after ingestion of the antimalarial agent primaquine. The degree of hemolysis can vary based on the dosage, coexisting infection, concomitant use of other drugs, or type of mutation. Exposure to naphthalene in mothballs and some herbal supplements have also been associated with hemolysis in some G6PD deficient individuals. Individuals with G6PD deficiency of classes 2 and 3 are asymptomatic until they take certain drugs. Hemolysis can occur abruptly within hours or gradually 1 to 3 days after drug intake, causing symptoms such as chills, fever, headache, nausea, and back pain. The severity of anemia can vary, and hemoglobinuria is a typical finding indicating intravascular hemolysis. Reticulocytes count increases within 4 to 6 days. Hemolytic episodes caused by infections are more common and can cause a drop in hemoglobin level if reticulocytes production is suppressed. Hemolysis induced by acute and subacute infection is not well understood, but the generation of hydrogen peroxide by phagocytizing leukocytes may play a role. Hemolysis due to infection usually resolves after recovery from the infection. Hemolysis in individuals with class 3 variants such as G6PD-A2 is self-limiting, while in class 2 variants such as G6PD Mediterranean, the hemolytic episode may be longer and not self-limiting. Favism is a severe and rare type of hemolytic episode that can occur in some individuals with G6PD deficiency after consuming fava beans. The symptoms of favism can occur suddenly within hours or gradually over a period of 24 to 48 hours. Hemoglobinuria, or the presence of hemoglobin in the urine, is one of the initial signs of favism. The severity of the hemolysis in favism can be influenced by patient factors such as the type of mutation, underlying disorders, and the amount of fava beans consumed. Only a small proportion of individuals with G6PD deficiency develop favism, and most of them have the G6PD Mediterranean variant. Neonatal hyperbilirubinemia. G6PD deficiency is associated with neonatal hyperbilirubinemia. Jaundice appears after 2 to 3 days of birth, without anemia. The main reason behind jaundice is inefficient conjugation of indirect bilirubin by the liver, not excessive hemolysis. Infants with G6PD deficiency and homozygous mutation in the promoter region of UGT1A1 gene are more prone to severe hyperbilirubinemia. Other factors like the type of G6PD variant and environmental factors also play an important role in the occurrence and severity of neonatal hyperbilirubinemia in G6PD deficient infants. Chronic hereditary nonspherocytic hemolytic anemia, HNSHA, occurs in a small percentage of G6PD deficient patients. HNSHA is characterized by persistent hyperbilirubinemia, decreased serum haptoglobin level, and increased serum lactate dehydrogenase level. Most patients with HNSHA are diagnosed at birth with neonatal hyperbilirubinemia and experience hemolysis into adulthood. HNSHA patients usually do not have hemoglobinuria, suggesting extravascular hemolysis. RBC morphology is unremarkable in HNSHA patients. HNSHA patients are susceptible to acute oxidative stress and may experience acute episodes of hemoglobinuria. The severity of HNSHA is variable and likely related to the specific mutation in the G6PD gene. Please don't forget to subscribe to our channel, activate notifications to get our new videos. If you like our content, please press the like button, and share the video with your friends. If you have any questions, leave a comment below. Laboratory findings. Hemolytic anemia. During a hemolytic crisis, anemia can range from moderate to extremely severe, and is usually normocytic and normochromic. G6PD deficient RBCs. RBC morphology is normal in G6PD deficient individuals, except during a hemolytic episode. Morphology during hemolytic episode. The degree of change in morphology during a hemolytic episode varies depending on the severity of the hemolysis. Severe variants may show marked anisocytosis, poikilocytosis, spherocytosis, and schistocytosis. Bite cells may be observed in rare cases of drug-induced hemolysis but are not a specific feature of G6PD deficiency. Bite cells are absent in acute and chronic hemolytic states associated with common G6PD deficient variants. Heinz bodies cannot be detected with right staining. They can be visualized with supravital stains, such as crystal violet, as dark purple inclusions attached to the inner RBC membrane. The reticulocytes count is increased and may reach 30% of RBCs. Serum levels. Consistent with intravascular hemolysis, the serum haptoglobin level is severely decreased, the serum lactate dehydrogenase activity is elevated, and there is hemoglobinemia and hemoglobinuria. The indirect bilirubin level is also elevated. The white blood cell count is moderately elevated, and the platelet count varies. The direct antiglobulin test, DAT, is negative, indicating that an immune cause of the hemolysis is unlikely. Tests for G6PD deficiency can be classified as quantitative and qualitative biochemical assays or DNA-based molecular tests. Quantitative spectrophotometric assays are the gold standard and require a hemolysate prepared from venous blood collected in heparin or EDTA anticoagulant. The rate of NADPH formation is proportional to G6PD activity, and the activity is typically reported as a ratio of the units of G6PD activity per gram of hemoglobin. Cutoff points to determine G6PD deficiency are usually set at less than 20% of normal activity, but this varies by method, laboratory, and population screened. Qualitative tests are designed as rapid screening tools to distinguish normal from G6PD deficient patients. The endpoint in qualitative tests is visually observed, and the results are reported as G6PD deficient or normal. Qualitative tests with deficient or intermediate results are reflexed to the quantitative assay for verification of the G6PD deficiency. The fluorescent spot test identifies G6PD deficiency by measuring the fluorescence of NADPH generated in the reaction. Blood and glucose 6-phosphate/NADP reagent are incubated and spotted on filter paper in timed intervals. Normal specimens appear as moderate to strong fluorescent spots under long-wave ultraviolet (UV) light. Decreased or no activity specimens do not fluoresce or display weak fluorescence compared to a normal control. Dye reduction qualitative assays use the same G6PD enzymatic reaction, but a second step is added in which the NADPH reduces a dye, giving a visually observed color change. An example of a dye reduction assay is BinaxNOW, which uses the enzyme chromographic test (ECT) method and has a sensitivity of 98% in detecting deficient specimens with G6PD activity less than 4.0 units per gram of hemoglobin. Another formazine-based test uses a tetrazolium monosodium salt (WST8) substrate, forming an orange-colored product. Dye reduction methods have an advantage over the fluorescent spot test, as they do not need UV light for visualization. Rapid, point-of-care screening methods that do not require venipuncture specimens are currently in development. Biochemical screening tests are effective in identifying hemizygous males and homozygous or compound heterozygous mutant females with severe deficiency, but they are not sensitive enough to detect mild and moderate deficiencies found in some class 3 G6PD deficiency and in heterozygous females. Moreover, they have subjective endpoints that may affect test reproducibility and accuracy. Reticulocytosis falsely increases the patient's G6PD activity over baseline values, leading to false results. Patients who are not G6PD deficient are expected to have high G6PD activity during hemolytic episodes. Hence, biochemical assays for G6PD activity should not be performed during acute hemolysis. Testing for G6PD activity cannot be performed after recent transfusion because the mixture of donor and patient RBCs does not reflect the patient's true G6PD activity. Moreover, WBCs and platelets also contain G6PD activity, and severe leukocytosis or thrombocytosis can affect the test results. In such cases, the buffy coat should be removed from the specimen before testing. DNA-based mutation detection is available in larger hospitals and reference laboratories. The DNA is extracted from WBCs isolated from whole blood, and molecular testing is straightforward as the vast majority of G6PD mutations involve single nucleotide substitutions. Rapid PCR-based methods can be used to target specific mutations with high prevalence in a particular geographic area, racial group, or ethnic group. However, if targeted mutation testing is negative, whole gene DNA sequencing is required. DNA-based testing is useful for prenatal testing, family studies, and identifying heterozygous females who typically have normal biochemical tests due to their mixture of normal and G6PD deficient cells. DNA-based methods can be used on patients who have recently received a blood transfusion, as donor white blood cells have a short half-life and will not interfere with the test. Additionally, these tests are not affected by reticulocytosis and can be done during an acute hemolytic episode. The disadvantage of DNA-based methods is that they require technical expertise and specialized equipment. Moreover, knowing the genotype of heterozygous females does not predict their clinical phenotype. Pyruvate kinase deficiency. Pyruvate kinase, PK, is an enzyme that plays a critical role in the glycolytic pathway of red blood cells. PK catalyzes the conversion of phosphoenolpyruvate to pyruvate, producing ATP. PK deficiency is an autosomal recessive disorder with an estimated prevalence of 1 in 20,000 in the white population, making it the most common form of hereditary nonspherocytic hemolytic anemia. PK deficiency is caused by mutations in the PKLR gene that codes for PK in RBCs and hepatocytes, with over 180 mutations reported, mostly missense. Homozygotes or compound heterozygotes with PK deficiency experience symptomatic hemolytic anemia. Some PKLR gene mutations are more common in the United States, parts of Europe and Asia. Pathophysiology of PK deficiency. The exact mechanisms causing hemolysis and premature destruction of PK-deficient cells are not fully understood. PK deficiency leads to a decrease in cellular ATP levels and an increase in 2,3-bisphosphoglycerate, 2,3-BPG. The increase in 2,3-BPG shifts the hemoglobin-oxygen dissociation curve to the right and reduces the oxygen affinity of hemoglobin, leading to greater release of oxygen to the tissues. This allows affected individuals to tolerate lower levels of hemoglobin. ATP depletion also affects the ability of the cell to maintain its shape and membrane integrity. Clinical manifestations of PK deficiency. PK deficiency can present with a wide range of clinical manifestations, ranging from severe neonatal anemia and hyperbilirubinemia requiring exchange or multiple transfusions to a fully compensated hemolytic process in healthy adults. Most patients have chronic hemolysis, including anemia, jaundice, splenomegaly, and an increased incidence of gallstones due to excessive bilirubin production. Rarely, PK deficiency can lead to folate deficiency, bone marrow aplasia, usually due to parvovirus B19 infection, and skin ulcers. Pregnancy carries a risk of fetal loss and exacerbation of anemia in the mother. Age-related iron overload and organ damage can occur, even in the absence of transfusions. The mechanism of dysregulation of iron homeostasis is not clear, but it may be related to a decrease in or lack of response to hepcidin, the major iron-regulating protein. Laboratory findings. Laboratory findings in PK deficiency include variable hemoglobin levels, reticulocytosis, anisocytosis, poikilocytosis, and polychromasia on peripheral blood film. Burr cells or echinocytes, in the range of 3% to 30%, are also observed, which increase after splenectomy. Howell-Jolly bodies, Pappenheimer bodies, and target cells may also be present on the peripheral blood film. Patients usually have laboratory findings of chronic hemolysis, including increased indirect bilirubin, decreased haptoglobin, and increased urinary urobilinogen. The WBC and platelet counts are normal or slightly increased. Tests for PK deficiency include quantitative and qualitative biochemical assays for PK activity, phenotypic assays, and DNA-based molecular tests for mutation detection, genotypic assays. The quantitative PK assay measures the rate of NAD formation, which is proportional to PK activity, using a spectrophotometer. More complex techniques may be necessary when variant forms of PK are suspected. Osmotic fragility is usually normal, and the direct antiglobulin test is negative. Qualitative tests are used for PK deficiency screening. These tests are based on the same principle as the quantitative test, where hemolysate and reagents are mixed and incubated. However, instead of measuring PK activity with a spectrophotometer, the loss of fluorescence is visually evaluated on filter paper to determine the oxidation of NADH to NAD. Mutation detection for PK deficiency can be done through genotypic testing. This involves sequencing the PKLR gene's exons, flanking regions, and promoter region. Thank you for completing the video. Remember to ask for ASCP short notes, and don't forget to subscribe. Bye.